Hello viewers, now we are going to discuss about production of ATP, or which is also known as phosphorylation. ATP is the energy molecule which is full form adenosine triphosphate. The production of ATP or energy molecule takes place by various metabolic process. And what is this ATP and what are the types of production of ATP? Let us discuss. Production of ATP takes place from ADP, for which ADP adenosine diphosphate having two phosphate. One inorganic phosphate is added. I means inorganic, P means phosphate. So it is represented as Pi, phosphate inorganic, is called as phosphorylation. So inorganic phosphate attached to ADP result in the formation ATP, that is called as phosphorylation. This formation of ATP takes place by two steps. One is called as substrate level phosphorylation and another one is called as oxidative phosphorylation. So in phosphorylation, two types are there: substrate level and oxidative phosphorylation. Let us discuss each in detail. Substrate level phosphorylation is the production of ATP where the energy is trapped directly from the substrate without the involvement of electron transport chain. That means there is no ETC occurring in this ATP production and is called as substrate level phosphorylation. That means when a reaction takes place, metabolic reaction, during that reaction directly the ADP is going to get attached to a inorganic phosphate, leading to the formation of ATP. Immediate release of ATP is called as substrate level phosphorylation. You can see in this we are going to depict two example, two reactions from glycolysis and one reaction from TCA cycle is going to depiction of substrate level phosphorylation. 1,3-bisphosphoglycerate in glycolysis cycle is converted to 3-phosphoglycerate. Here ADP is converted to ATP in presence of enzyme phosphoglycerokinase. Kinase means kinetic, means kinase, kinetic energy. So here energy is involved, kinase. In the starting and ending compound you can see the common name phosphoglycerate. So 1,3-bis phosphoglycerate and 3 phosphoglycerate. So in both the names start in the substrate and product you can see phosphoglycerate, that's why name phosphoglycerate kinase. So it is a substrate level phosphorylation. ADP is converted to ATP directly. And in the second one, phosphoenol pyruvate, this also from glycolysis converted to pyruvate, and ADP is converted to ATP. Phospho after that enol pyruvate. So the first word phospho is there in the substrate. After the arrow mark or in the product you can see pyruvate is only is there, phospho is word is not there. That means the phospho present in the phosphoenol pyruvate is removed and attached to ADP to form ATP in presence of enzyme kinase, and this reaction takes place with the help of pyruvate, so hence pyruvate kinase. Succinyl CoA is converted to succinate. This is from TCA cycle or Krebs cycle. GDP is converted to GTP. Instead of ATP you can see here GDP in presence of enzyme succinyl thiokinase. So ATP and GTP are very much similar, uh, and GTP can be converted to ATP. So this is substrate level phosphorylation where energy is produced directly with the production of ATP. Substrate level phosphorylation. Next is oxidative phosphorylation, production of ATP when where the energy is trapped from the oxidation of reducing equivalents such as NADH and FADH in electron transport chain is termed as oxidative phosphorylation. During metabolic reactions, whenever a pathway or cycle takes place, in that NAD or FADH produced in any step, they are indirect form of energy, and they have to be converted to ATP that takes place or occurs in electron transport chain. Whenever NADH or FADH enters electron transport chain, they are converted to energy molecule, that is ATP. One NADH can lead to formation of 3 ATP, and one FADH is equivalent to 2 ATPs. So NADH plus H plus gives 3 ATPs in ETC, and FADH2 gives 2 ATPs in the ETC. So this is the indirect form of energy. That means it has to undergo the ETC process so that we can get energy molecule, that is ATP. Hence it is called as oxidative phosphorylation. Oxidation means removal of hydrogen. So you can see here NADH when it goes electron transport chain, or FADH goes through electron transport chain, the H is removed and that H is used for utilized for production of ATP. So H is removed, so oxidation takes place to produce ATP, hence it is called as oxidative phosphorylation. Requirement for oxidative phosphorylation is reducing equivalents like NADH or FADH. Electron transport chain present in mitochondria. So this electron transport chain or oxidative phosphorylation process takes place in mitochondria. Next we are going to discuss about glycogen metabolism. So what is glycogen? All of you know glycogen is a homopolysaccharide made up of same type of sugar units, that is glucose, present in animals, whereas plant stores starch, animals stores glycogen. And glycogen is having a branching at every 8 to 10 glucose molecules. Glycogen is an animal storage polysaccharide. It is synthesized during the excessive glucose in the body, that is converted to glucose, and it is stored in the muscles and liver. And breakdown of this glycogen is called as glycogenolysis, and it takes place during decreased glucose level whenever the person is doing fasting or not having food. In that condition, to provide glucose for the blood, the glycogen storages organ is undergoing breakdown of glycogen to produce glucose. That process is called as glycogenolysis, whereas the production of glucose when glucose is more, that is called as glycogenesis. So in the first line you can see it is synthesized during fed condition, means whenever you have a very fed good food, and broke down glycogenolysis takes place during fasting, that means whenever you are not fed or when you are in a hungry or in a fasting condition or starvation condition. Glycogenesis let us begin with, that is also called as glycogen synthesis. Genesis means generation, genesis means generate or synthesis. Glycogenesis means synthesis of glycogen. So definition tells formation of glycogen from glucose is called glycogenesis, and hence glucose aromer glycogen is glycogenesis, where in it takes place in liver and muscle. In the liver cells and muscle cells, glycogenesis takes place. Which part of the liver cells? Because cells contain different parts: mitochondria, endoplasmic reticulum, Golgi complex, etc. So where it takes place in cell of these tissues, it is in cytosol. So what is the starting compound? As the definition you can see, formation of glycogen from glucose. So starting compound is glucose and end product is glycogen. This is about introduction part of glycogenesis. Let us discuss the pathway, how the glucose is converted to glycogen. You have seen the starting material is glucose, ending material is glycogen. So this pathway you can see here, the starting beginning is glucose and ending compound is glycogen. So how this takes place? It takes place through several steps, and this stepwise process of conversion of glucose to glycogen is called as glycogenesis. Let us discuss step by step. In glycogenesis, the glucose which is present in excess is taken up by liver cells and muscle cells. They are going to convert this glucose to glycogen in the stepwise manner. In the first step, glucose is converted to glucose 6 phosphate. Here the phosphate is added, and to form glucose 6 phosphate, ATP is converted to AMP. ATP is converted to ADP in presence of glucokinase or kinase, and then this glucose 6 phosphate is converted to glucose 1 phosphate. Glucose 6 phosphate means only one phosphate attached to the sixth carbon atom. That phosphate attached to the sixth carbon is shifted to first carbon atom, that is mutation takes place. Phosphoglucomutase takes place. So glucose 1 phosphate is obtained. This glucose 1 phosphate combine with UTP, uridyl triphosphate, to form UDP glucose with release of pyrophosphate PPI in presence of enzyme UDPG phosphorylase. The enzyme's name is not shown in the slide. You can go through the audio and then you can ensure the enzymes names. And UDPG glucose is a glucose carrying molecule and it's going to attach this glucose to a small chain of glucose, which is called small chain of glycogen, that is glycogen primer. If 5 glucoses is already attached, that is called glycogen primer. This one glucose which is with UDP is going to attach to that to form 6. So like that, one by one glucose is going to be attached by UDP glucose to form a long chain of glucose, which is going to form glycogen. So UDP glucose is by addition of glucose, one glucose in the fourth position, alpha 1 4 glycosyl units are obtained by presence of enzyme glycogen synthase. And since glycogen is having branching for every 8 glucose molecules, this alpha 1 4 long chain linear straight chain glucose polymer will undergo branching at every 8 glucose molecules by an enzyme called as branching enzyme. Thereby you will get a product alpha 1 4, that is the straight chain linkage, and alpha 1 6, that is the branching chain linkage of glycosylates takes place to form glycogen. This process takes place elongation of both linear and branched chains, and a larger molecule of glucose polymer, that is glycogen, is obtained. This takes place with the help of branching enzyme glycosyl alpha 146 transferase enzyme. This is about glycogen synthesis. So significance. What is the significance of glycogen? Glycogen is an animal storage polysaccharide. It is stored in the liver and muscles of the animals. When blood glucose is less, then to maintain the glucose level, whatever the stored glucose in the form of glycogen is converted back to glucose, and thereby it maintains the glucose level in the body. This glycogenesis is can be regulated. Some regulatory enzymes are involved for regulation of glycogenesis, whether to start the glycogenesis or to stop the glycogenesis. The certain regulatory enzymes are involved in that glycogen synthase can be inactive form or in active form. Hence, the glycogen synthase is going to be a regulatory enzyme for glycogenesis. This is about glycogenesis. Thank you.